Okay. Let's pay attention, please. So we're going to start with mycology 1, and that's what we used to teach in terms of human physiology, survival of the fittest. And when we talk of mycology, interestingly, it changes. So we talk of survival of the prettiest. So let's see what does it mean. There's a lot of studies done, and I mean, you can read it on your own. But the most important thing is that this is where the term is coming from. One of the psychologists, he wrote this book, and I want to point out one of the English term, fungible, like fungible asset, that we use this term fungi or fungible to mean something, and that actually should kind of give you an idea. If you go into literature, the English term means movable, perishable goods of a sort that may be estimated by number or weight, such as grain, wine, etc. So probably in older time, they used to kind of trade off in some of the things that are fungible, so we can exchange. The reason I'm talking about that is that I'm pretty sure you are a pharmacist, and you don't have like a tunnel vision, so you know what's happening in a pharmacy. And I was talking to our P4, Vince Ransford. How many graduates pharmacists per year? More than 14,000, 15,000 per year. So by the time you graduate, you can imagine what's going to be the market like. So these are some of the economical things that you have to keep in mind. Don't be a tunnel vision. Just I'm going to go there and Walgreens are going to give me a golden handshake and I'll be the manager and so on and so forth. The important thing in this aspect is that if you look at pharmacy and the drugs, and if you happen to work in a pharmacy store, is that there are people who calculate their moves, the people who plan, give career advices, and give advices to people who are going to look for residencies and so on and so forth. And what I meant by the survival of the prettiest was because $400 billion are being spent on cosmetics. And you wonder, what has it got to do with mycology, and what has it got to do with the subject I'm going to talk about? Interestingly, a couple of years ago, and this is a fact as well, this is our culture as well, American culture. If you want to spend more time, more money, more whatever, than education. Good for us, no problem. So if you work in pharmacy and you want to break down the things which are there, those people who run our lives. I don't know how many of you did watch Democratic debate yesterday, but some of things are good for you to know in terms of where the government planning, where the money's going, all these things are interesting. So they may give you a financial responsibility that where you're going to move forward to. I should give an example of one of my students right from the P1, right? He was asking me, how would I do an MBA? So as we talk about PharmD, he said, forget, I'm going to do it anyway. I'm going to do PharmD and then do MBA because I'm going to go for business. So sometimes people are very clear into what they are looking forward to, right? But I think in this day and age, if you look at the global industry, so you will see the amount of money being spent on cosmetics, okay? Not only in this part of the world, but basically, so if you look at sales of some of the creams, for example, it's going to grow and the need will grow as compared to, for example, antibiotics. If you teach you antibiotic, I spent like 10 lectures of penicillin, is resistant. You learn all these carbon cycles and all these All those things that are there, and by the time you graduate, it's no longer in use. But some other aspects are growing. Some aspects are there which have a market there, and people who plant, that's what they do. Okay? So anyway, keep this in mind as kind of a building block. We're going to start with fungal classification, structure, and replication. Fungi in the real world give color to this world, right? They have amazing color. They are beautiful. They are very well organized. They are very well organized, and as you must have noticed, viruses are also very well organized, and they also cause us problem as well. So if somebody was to see this as very well organized, colorful things, fungi, and in the color world, even in the real time, you can see that these basically happen to be the most beautiful structures in nature. So fungi happen to be the most beautiful structures in nature. So you can see from different colorful things, coral leaves, deep sea, you name it. So everything we already in terms of color in the nature, the chances are it's the fungi. They are very complex, right? They are multicellular, and they are colorful, okay? Now, having said that, and with 400 billion budget on beauty, most of the time, we spend it on skin care, hair care, and nails care. I'm not going to go and break up how much an average teenager spends on that, but you know that people, there's an industry there, there is spending power in the people do care, and so do the pharmacists, and so do we. So what I'm going to do is tell you that most of the problems that you get in taking care of these things, and most of the over-the-counter things that are available, the enemy in all the cases is nothing but fungus. So you're going to lose your nails because of the fungus. You're going to have all those problems because of fungi. You're going to have loss of hair, thinning of hair, breaking of hair, two edges, what you name it. And the reason is behind that is fungi. So you need to get a really good hold of fungi. So interestingly, FDA does not regulate cosmetics. It's not going to tell you it's going to cause cancer, it's this and that. None of their business. You make something, you can bring it, sell it to Walmart, Walgreens, they will bring it in. Interesting, right? And people will apply all the things on their skins, on their hairs, on a daily basis, on a weekly basis. But at the same time, antifungal drugs have to be prescribed. Antifungal drugs have to be one of the most expensive drugs in the market right now. Other than small percentages of antifungal creams and so forth, the real drugs that we're going to talk as a part of mycology is for your interest, that you need to know, because you're going to be in this situation that people will come advise for you about the skin care, hair care, and the nail care. And that's what this subject is about. So I'll talk about the importance of fungi, which I think I've tried to already give it to you in terms of economic burden, in terms of need of the people, in terms of something which is going to come up. So all those cosmetics may be the best-selling drugs of the future because of the care people take, in terms of managing, that purifications. So that's pretty much is there. A lot of behavior issues, so on and so forth. But this is out there. We're going to talk about classes of fungi. I'm going to talk about structural replication, difference between yeast and mold, metabolism, reproduction, and clinical feature in human diseases caused by fungi. So we'll see how we move. And these basically is not only today's lecture, but pretty much the same objective for all the 5 lectures that I'm going to give in this branch of microbiology. Now, pure microbiology, fungi are present everywhere, ubiquitous, very diverse, and you wonder what are they doing in nature. So if you were to ask, a botanist or a biologist or a very basic scientist, the main purpose is to degrade organic matter. We need that. Otherwise, the dumps and dumps of garbage and everything which is there, smelling, rotting. So that's pretty much the purpose. And imagine if you get infected with that, so they will start degrading you as well. And many times you can notice that if you have athlete's feet and you open up the finger web and you see what is happening in the skin, right? Is dead skin, is causing pain, is redness, whiteness. All these things are there in terms of the diseases that it causes. Now, these are heterotrophic existence. This means that we basically think of 4 different aspects of pure microbiological survival: saprobes, the organisms that live on dead and decaying matter; symbionts, we talked about that earlier, that live together in which the association is mutual advantage. Sometimes we talk about that in commensal bacteria. Commensals, again, a close relationship. One benefits from the other, and the other neither benefits nor is harmed. So that's more of a thing in terms of commensal, unless you give them an opportunity to poke a hole on your skin or cut your skin or abrade your skin and cause them to enter. Parasites, when they live within a host, derive benefit and in turn cause disease. So that happens especially for viruses as well. So these, by definitions, we call them pathogens because they cause disease. Now, the other important thing, keep in mind, general question, you cannot give antibiotics to treat fungal infection. That's a major question I'm going to ask you over and over again. And there has to be a good reason for that. Now, the reason is fungi are eukaryotic. Who else is eukaryotic? We. So this means whatever you're going to give it to them, it's not going to kill fungi, but kill you as well. So that's a challenge there. They have true nucleus. They have a ribosome ADS. They have mitochondria. They have endoplasmic reticulum. As compared to bacteria, which are prokaryotic, no nucleus, 70 S ribosomes. All those antibiotics acting on the ribosomes cannot be acting over here. They don't have mitochondria. They don't have endoplasmic reticulum. Major difference. Now, what has happened over the past few decades is that they have become a cause of major disease, especially in immunocompromised and hospitalized patients. Hopefully, if your immune system is good enough, you should be able to take care of fungi. But if your immune system is messed up, then you have a problem. So they can serve as opportunistic pathogen, and they cause considerable disease and death. Don't think that they are that naive. They cause big problems. To such an extent that the incident of specific invasive mycosis, so you remember the way I presented like cosmetic, that's not the end of the story. That's what I wanted to imply. That's what people get worried about. It's so obvious. People look at the skin, people look at their hair, people look at the nails, something they look at and perceive, and they want others to look at it. That's like a beautification. But the fungi can penetrate the skin and mucous membrane and invade, called invasive mycosis. And you heard the story, I think, a couple of years ago about fungal contamination of some of the drugs or some of the injections in upstate, in the northern states, because there's big litigation there. So fungi, again, can contaminate injections as well. The important thing that you have to keep in mind is there are no non-pathogenic fungi. Right? So this is like a general message that you have to keep in mind. I don't want to emphasize more than what I already did in terms of AIDS and fungi, because remember I said the most of the time the presentation is oral thrush. So they got some of the presentation in recent years because of immune system and disease immune system, these are the first signs of immunocompromisation, like AIDS. So they present with that. And the other important thing is that if you change the ecosystem or change the normal flora or remove that good sets of bacteria by giving antibiotics or long-term antibiotics or inadvertent use of antibiotic, you are going to give chance to this fungi, which will come and take over. Okay, that's another important issue. And then again, like bacteria, if you have invasive procedures, you will end up getting fungi into the system, and then they will do the rest. Now, in terms of classification, taxonomy, they have their own kingdom. Kingdom fungi. We already talked about eukaryotes. The most important thing that I'm going to discuss in the next few slides is that still having said that they are eukaryotes, but they are distinguished from us, for example, because had it been the case, we would not have any antifungal drugs at all. Right? So there's a little bit of difference, and you want to go and explore that so we can use drugs that will save you and kill fungi. So good thing is, in eukaryotic kingdom, the eukaryotics, especially these one, have a rigid cell wall, and that's composed of chitin and glucan. Two important things. So you can imagine, I don't have to tell you that you're going to go after these two. You're going to go after these two because they are different than us. And the second thing is that they have a cell membrane in which ergosterol is substituted for cholesterol as the major sterol component. So they have a cell membrane where normally we have ergosterol, they have cholesterol. So that's another important thing in terms of understanding the antifungal therapy. Now, if you look at the typical structure of a typical fungi, nucleus, mitochondria, and the plasma reticulum, they have cell membrane over here. I said cell membrane, right? This cell membrane has ergosterol and not cholesterol. So you can imagine if you want to go to kill this fungi, you're going to go after this. The other important thing is that they have a cell wall. You can see the cell wall over here. And I said that the rigidity in terms of the structure element, they have chitin and glucan, right? And then the other important thing is cell wall mannan linked to surface protein. So that's another important thing as we discussed. And I would want you to review the differences between gram-positive and gram-negative, and also mycobacteria when we talked about mannan group, lipomannan, lipoarabinomannan. So that mannan group, especially in gram-positive, as well as other, gives you the structural kind of an idea as to, especially, I would want you to look at the mycobacterium cell wall, where you see the outer structure are deeply anchored to the inner protein. So that kind of keeps them in the track. All right. Now, they have a structure we taught polysaccharide, like a capsule, and good for fungi because the function of that is, of course, is anti-phagocytic and is a virulence factor. Okay. So, and capsule yeast, when we talk of some of very specific capsule, for example, in some of the subtypes of fungi, we talk about Cryptococcus neoformans. Who did that as an individual project? I know it was one of the projects to the students. Very interesting. I've been asking this question over and over again. So I want to make sure that you've done what was supposed to do. Very interesting. Okay. Right. Cell wall, again, like any cell walls, is an antigenic in nature. It's multi-layered. As I said, you want to compare gram-positive, gram-negative, and tuberculosis. Polysaccharides, 90%, hexose and hexamine polymers. Proteins and glycoproteins are 10%. The function, of course, like any cell wall, is to provide the shape, rigidity, strength, and protection from osmotic shock. So this is how fungi have survived over time in terms of whatever they are doing. Last year question, I don't know whether you added this as a question or not, Answer? You add this as a question, so I had to answer that. So you can see that for microorganisms, size does matter, right? So because I told you about the membranes, I told about the invasiveness. So you can see that this is a question from the last year. And the answer is more or less the size of a virus. It's like small size. So again, if the size is small, it can reach to places where the others cannot reach because of the size. If you look at the journal Fungal Taxonomy, if you look at their morphology and look at their structure, biochemical, molecular, all these kind of different characteristics, as we will do over time. But since I'm starting off with a very general overview of fungi, what I want you to, at this level, appreciate, and I will also advise you to start reading those PDF reviews, because they have been copied from a book that gives like a structural element in terms of charts. So you can visualize when I speak what is going on in terms of different branches of fungi and what clinical pictures are there. Concept map, it's called concept map. So fungi may be unicellular or multicellular. The most simple grouping that we normally do is based upon morphology. Either they exist as yeast or mold. That's like a general layman terms, and we still want to use it. Okay. Now, having said that, there's another term we use in fungi. It's called dimorphic fungi. Dimorphic, as the name suggests, they have two morphologies, and interestingly, they are fungi because we eat fungi, right? What do we eat? Mushrooms, right? So of course, they should not be included in this category, but most of the time, the fungi of medical importance are dimorphic. The reason is they exist in both yeast form and a mold form. So that's the one that we're gonna spend more time in terms of knowing. So yeast is a unicellular, and it reproduces by budding or fission, where there is a mother cell and you pinch off daughters from the mother. So that's a typical budding, and I'm pretty sure that you would have seen that. And what may happen when the daughter cells are bud off from mother cell, and it's like speed up process. So you may see a different kind of a version, as if they are segmented. They are hyphae, like, you know, attached to each other, but they, in the real time, they are not. So such a morphology we call pseudo hyphae. We call a pseudo hyphae, like a sausage, like a penis. I'm going to show it to you. Most of these are round, pasty, and mucoid colonies on agar, and we use in baking, right? Right? Okay. Now, this is nuclear fission, dividing into two, budding. But you can see that they are budding off, and it's so fast a process, they appear that they are like attached to each other because pseudo hyphae, but they are not. Now, this slide will give you an idea how would you differentiate between yeast and mold. So this is a yeast, unicellular, as compared to mold, which is multicellular. So M for mold, M for multicellular. This is an old mnemonic, so you don't mess that up. Okay, so these are multicell, they are real hyphae. They look hyphae, but they're not. That's why we call them pseudo hyphae. Okay, and then again, they go to different phases. So we'll talk about the coenzytic hyphae. Some of the hyphae are separated by this kind of clamp connection, and some of them may have different morphology. So you can see from here, they all are molds, but they may appear in more than one form, and we're going to discuss that. But this slide will just give you an idea difference between yeast and mold. Sometime what happens is that when one of the yeast is budding off, so that budding off is kind of stretched. It's kind of gluey, sticky, mucoid. It's kind of stretched off. It looks like a germ tube. It looks like a germ tube. So that's characteristic of a yeast. If you were to look at infection, we do like skin scrapings. Can somebody close that door? Because we have offices over here. Whenever the doors are open, we can't even read in our offices. The voices are too loud. Okay. So if you were to look under the microscope and this picture appears, yeast. If this picture appears, mold. But I already told you the problem with human infection with dimorphic, because they can exist in both. Okay, now molds, as I said, M for molds, M for multicellular. They have hyphae, and they elongate at the tips by a process known as apical extension. And these, I told you, two different types of them. One are co-anocytic, which are hollow and multinucleate, as compared to septate, which divide by a partition or cross walls. You can look at the picture, you'll see that. So the hyphae, they get together and produce a mat-like structure, which you saw. We call it mycelium. We call that mycelium. So these are some of the terms, and I think they may be in the glossary of your book as well, and the medical terminology terms that we normally use when we talk of fungal infections. So naturally, one of the questions that you will always be asked, what is the difference between mold and yeast? I already told you, single cell reproduced by budding. Molds have hyphae, filamentous, and they grow by branching. So it's more of like a tree-like pattern that you normally see. Now, the other important thing in terms of the aerial hyphae, which produce conidia, that's another thing. Let me see if I can enlarge that. This is an example where they're going to divide as sexual or asexual way. So if you look at the example of asexual way, which is more, let me see if I can enlarge that. You can see from here, like a typical botany thing, when you look at the plants. So you will see that you will have a branch. It looks like a plant, and then you have a sporangium, like we talked about some of the parasites. So sporangium, and it has those small little dots, spores. Okay, we talked about spores, bacterial spores, but they were in different contexts as compared to this one, right? And if you know a little bit of botany, like a flower petal with male and female, with the uterus and sperm, pretty much the same thing. The sperms going to fertilize the eggs within the flower. So for the pollination, same concept over here in terms of fungi. Okay, and if you compare that with the other one, which we call, this is like a zygomycete. Again, the difference is that one is spores with a membrane, like a ball membrane. The other is conidia. So these are like conidia. So these are conidia and spores, which are two different forms. And as we move along, I'm going to explain that to you, that, for example, spores are characteristic of one of the very specific and dangerous fungi that we see in clinical practice called zygomycete. So zygomycete has spores, as compared to conidia, where we have aspergillus. So aspergillus, this is a kind of another fungi. And you can see from here, what's going to happen is that they will be blown out in the air. So you can inhale. So that's another important thing. So you keep that in mind in terms of transmission of these fungi that we normally would see in terms of, especially for the conidia, we call them aerial hyphae. The reason is they're going to be spread out in the air. Okay, and that's mostly asexual reproductive elements that we normally see in this kind of fungi. So one was the difference between bacteria and fungi. The other important thing, I mean, the way we are discussing right now, that you see a lot of familiarity with the plant as well. Okay, so in this case, we want to differentiate between fungi and generally plant. The most important thing is the fungi lack chloroplast, and they do not have photosynthetic energy producing mechanisms as the plants have. Otherwise, if you get implanted with a fungi, it's going to grow a tree in you. So we'll have another problem to take the tree out or plant out. You know, so that's good for us. Now, when we talk of colonies, because remember we talked about bacterial colony, shape, size, and everything, and I'm pretty sure you must have seen that, you know, when you're going to leave your bread on the table or not going to attend to whatever you put in the refrigerator, the mold comes, right? Or you see that colorful thing, and if you were to look at it very carefully, so you will see that sometimes it's filamentous, hairy, or woolly. If you were to grow on agar or agar, it's going to produce very typical vegetative hyphae in the culture medium. And as I said earlier, you have to look under the microscope and you can visualize what was depicted in this picture in terms of what is going on, and they will look like plants. Okay. Now, one of the other terms that you will see that we normally talk of is that budding. We talk about the budding, but budding was different for viruses. Budding is a little bit different over here. So the conidia that you saw in the previous slide is produced either by budding, or they have, in budding, basically what happens is they have these segments, and all those will fragment and form individual cells. We call conidia or orthoconidia. I'll discuss the difference while we move along. The most important thing is that these conidia can easily be airborne. So if you're running in the field, or if you're there, if it happens to be molds in that area, or you're living in a house which molds is a serious thing, you're gonna take it in and cause respiratory problems and fungal diseases. Okay. Now, what we do is that we take the sample and put it up on agar and look at the size, shape, because they're all different. And I'm gonna tell you right now, as I will take one fungi at a time and give you the morphology and detail and how does it look like under the microscope, but I'm gonna ask you to compare it. So all those concept maps that are posted on Moodle have like tables over there. So you have like 6 different fungi, and you are comparing one with the other in terms of morphology. Okay, and some of these morphological questions will just appear as you have bacterial spotting as well. So it's very clear, you have the basic, you should be able to identify, and that's important. Now the second thing is we want to look at their metabolism because we want to use antifungal drugs. Okay, most fungi aerobic respiration, though some may have facultative anaerobic, so they can ferment as well, and some are strict anaerobe. So basically, you can see from there all the way from aerobic to anaerobic to strict anaerobe. So these are the 3 important aspects, and we already spent a lot of time on finding out oxygen consumption, because this oxygen is going to go and be processed in the electron transport chain, the Krebs cycle for energy and production of ATP. Okay, last year question. You can see this is the question came last year. And again, All of them are true. Most fungi, I just kind of copy and paste from the previous slide. You can see from here, aerobes, some can go anaerobic. You can see a few anaerobes. Anaerobes are not human pathogens. That's something that is like a fact, right? So anaerobes are not human pathogen as compared to the others. Okay, now when we look at their diversity that they have in terms of versatility, in terms of production, and also keep in mind that bacteria, and remember when we taught you bacteria, we talked about Alexander Fleming, and what he found was mold, penicillium, that could kill bacteria, right? So don't think that fungi are of not good use. So they have a good use. For example, you can see that all that citric acid, ethanol, glycerol that you're seeing, even penicillin and other things, we are producing and we are getting it from fungi. So they are producing that. And we make industrial use of that, colors as well. But at the same time, we can use the same thing when it cause problems in terms of diseases that, and pathophysiology. But the important thing is that relative to bacteria, fungi are slow growing and doubling time is hours rather than minutes. But also keep in mind, many times for treatment as well, they need long-term treatment. You must have witnessed yourself, athlete foot takes ages, sometimes it doesn't even go away at all. And many times people who have athlete foot to begin with, they will end up having a nail infection as well. And once it gets to the nail, then it's very tough to remove, So you can imagine the chronicity of infections in these cases. Okay, now let's begin with fungi of medical importance. And as I said, in the 5 lectures, I'm going to talk and take them in detail. And we talk about antifungal drugs, but don't get scared by the names. But if you pay attention, all of them have mycetes in the end. Mycetes. So mycology, fungi, simple. Zygo, Archiasco, Basidio, Hemias, and Uesco, like Mexican, right? Maybe it is, I don't know. But these are some of the terminologies. Tons of mnemonics, year after year, students make it. We need to know them. Now, when we talk of the other important thing, as I said earlier, when we talk of sexual and asexual reproduction, pretty much mean what you must have done and I must have done ages ago in botany. Same plant thing, mitosis, meiosis. I don't want to go in detail. And all those terminologies that we know in terms of cell splitting, and all those terms that we use in terms of teleomorph, anamorph, all these terminology holds good for plants, it holds good for binary cell division, it holds good for fungi as well. Okay? Now, the problem that we have is we need to diagnose that. You're going to collect a clinical specimen. Remember, that was something I talked about just before that last lab, that we have to get a clinical condition we want to treat. So what is it that we're going to get? So the clinical specimen, the fact is that even if a fungi has an ability to produce sexual spore, in clinical condition, it is common to refer to asexual designation. The reason is that we tend to isolate anamorphic or asexual most of the time when we take the specimen. Okay? And so what I'm trying to say is asexual is something that's so common. But if you want to have the same fungi to produce sexually, so you need to provide them with culture media, right? So you could provide them with the culture media. As you remember in parasitology, we talked about diagnostic stage of those parasite and we talked of infective stage of parasite, they were different. Sometimes you can diagnose a particular component of the cell cycle, but they may not be infective. So that's something you have to keep in mind when we talk of a clinical specimen, when we talk about sexual and asexual. Most of the time, the isolates in clinical specimens are asexual. Now, if you are going to ask me how many of all those 5 classes that I talked about, there are thousands and thousands of fungi. Good for you. You just have to worry about only 200. Okay, but again, things are increasing, but they lump into the 5 major category, and we treat them accordingly. So regardless of what is going to happen, we kind of treat them as one, lump them in one group. Okay, so we are more interested in picking up those classification differences, and I'm going to give you the clinical classification as well. That probably I would expect you to know and not to remember all 200. So this is what I think I decided that I'm gonna, other than the major classification, I would hold you responsible, and I'm gonna take it from here. And this is also very interesting that most of the drugs which are available are gonna go over here. So keep in mind human mycosis, and this classification is based upon invasiveness in terms of penetration of the skin and mucous membrane. Okay, because these are the barriers that we have, natural barriers. So superficial mycosis, cutaneous mycosis, and subcutaneous mycosis, I don't need to tell you that these 3 are pertaining to skin and mucosa. Okay, all simple natural barriers of immunology, all simple general rules of pathogenesis hold. Okay, when I said to you that you give them an opportunity accidentally or incidentally, by abrasion, by cutting, by any other mean, so they will go and take advantage of that. Okay, so keep that in mind as general rule for bacteria, viruses, and fungi as well, right? And I'll have maybe one lecture tell also skin, since they are involved in skin and mucous membrane. So for STIs, we talked about gonorrhea, we talked about syphilis, the major ones that we have in terms of bacterial STDs. And when we talked about viruses, we talked about human herpes simplex, we talked about papilloma, and what was the third one? Hepatitis, okay. Well, then 4th one. AIDS, we talked AIDS, retrovirus. They are all considered as STIs. Now, likewise, they are also considered, according to CC, because of the invasiveness, because of the opportunity that is provided to them accidentally, incidentally, to cause problem. That's why I said yesterday, or whenever I was talking about the AIDS, that human secretions have these viruses, and many of the things that happen in terms of needle skin injury, tattooing, piercing, or anything of that sort will give them an opportunity to cause infection, may it be bacteria, fungi, or viruses. Is a natural phenomena, okay? So keep that in mind because it's going to go over and over again. And that's the mechanism of pathogenesis for all microorganisms. Endemic mycosis means that it's going to go and penetrate into your body. So your liver, brain, spleen, any part of your internal organs can also be infected. Okay? And then opportunistic mycosis, I just gave you an example for that. Okay? That makes sense? So you just want to relate things with each other, natural barriers for skin and mucous membrane, and anything that goes along. Okay, now the problems of therapy for fungi is because they are eukaryotic, biochemically similar to human host. It's difficult to develop chemotherapeutic agent that will destroy the pathogen and not harm the patient, is a rule. But again, if you understand what I just said, differences between human and mycology in terms of chitin and glucan, I thought about ergosterol. These are important because the drugs are going to be targeted towards them. Now, as you saw that clinically, we classify them as cutaneous, subcutaneous, and systemic. Cutaneous, of course, skin and mucous membrane, mostly. What was the typical presentation of AIDS patient? I just said a while ago as well. How do they normally present? Thrush. Thrush. What is thrush? Oral candidiasis, oral candida infection, oral fungal infection. Okay, because of the immunity. Okay, so keep that in mind, that your immune system goes those, you may have a problem. And cutaneous, subcutaneous, systemic depends upon how invasive and penetrative these fungi are. Okay, and then you may wanna, I'll see if I may have a slide from your skin. So you have to know the layers of the skin, because some fungi are gonna be on the top, some in the middle, some on the bottom. And then again, it will be easier for you to understand how are we going to treat them. The other important thing for fungal diagnosis is that we, as I said, that you need to know and visualize that. So we take the skin scraping or mucosal scraping or specimen and look under the microscope. What is the commonest fungal test done in the world? Okay, I didn't say the US, in terms of looking for a fungi. What do you think will be the commonest test done in the world to look for fungal infection? Or let me make it your easy, yeast infection. Which test? Then serological test mean antigen antibody, not. No, name the test. Sorry? Pap smear is for cancer. Think simple. We say that think simple, think green. That's what means mycos. Commonest test, yeah. Test examination is not a clinical test. I'm talking about microscopic examination. What is the commonest microscopic examination done to diagnose fungal infection? Sorry? Okay, I'll give myself one credit. Vaginal smear. In CDC things, there's not a single woman who has not have fungal infection. It's so common for the commonest test done as a vaginal smear microscopy. They look for fungi because vaginitis, fungal vaginitis, is the commonest disease occurring, and that's 100% in the lifetime of a woman. So that's the commonest test done. And we'll talk about that because that's also a part of STI as well. And yeast infection. So that's the most commonest over-the-counter drug given, and those on the pharmacists will tell you that's the yeast infection in women causing vaginitis. Anyway, so culture can also be done, and serological tests again can be done, antigen antibody tests, and we can also do DNA probe, that's sure. When we talk of culture, you just have to remember one culture, Sabouraud's agar. And you can bet it's going to come. Sabouraud's agar is the only media, but the only catch over here is when you want to grow a fungi, what will happen is, so you want to have a low pH because you want to inhibit bacterial growth. So sometimes we do, so the chances were, as it happened in Alexander Fleming, he didn't want to grow the mold, he was working in bacterial cultures, and it got infected with molds. So if you want to grow molds, it's going to get infected with bacteria. So you may want to add a little bit of antibiotics over there sometimes to make sure that you only grow. And why do you need to culture it? Why do you need to culture fungi? I just say size, morphology, antibiotic sensitivity, all those things that we talked about in bacterial, same goes for this one as well. And this may have happened if you were to leave fungi. So that's what happens. You can see this, the shape, size, color, odor, all these things when they go and test and grow this fungi onto the saborod's agar. Okay? So I'm going to take questions from you, and